So in the last episode, I talked about how when you're in reductive stress, you don't want to drive superoxide production when you have a lot of NADH hands its electrons to the electron transport chain at complex 1. The electrons move through the electron transport chain and they come down over here and they make ATP. And that's great when they do that. But what you don't want is NADH. We're going to go over it. It's going to be fine. So this is the citric acid cycle. This is how we burn all of our calories, essentially. This is how we get this acetyl-CoA, as does alcohol, as do many amino acids. And the acetyl-CoA, or the acetyl group, really goes through a bunch of reactions. And you see these ones underlined in red. So it requires an NAD plus at this step, and the NAD plus gets converted to NADH, and the NADH is going to carry those electrons up to the electron transport chain to make energy, to make ATP. The same thing happens at the next step. You need another NAD plus to keep going, and you need another NAD plus to continue, going around and cycling. But you get another NADH out, and all those things are going to make ATP. But right here at this step, and this is succinic acid, which also known as succinate. So here you were also taking electrons. So really just these high energy electrons that live in the bonds between carbon and hydrogen. And you've heard of hydrocarbons before. Obviously oil, when you burn it, gives off a lot of energy. It's a hydrocarbon, and that is just because the bonds between the steps, what we're doing is we're harvesting the energy stored, the calories stored in those electrons, right? And so there's two different ways we do it. Most of the time, FAD, and this is the case when succinate becomes fumarate. FAD is the electron carrier, and when it has the electrons, it becomes FADH2, which is the reduced form. NADH is the reduced form. FADH2 is the reduced form because you're reduced when you have the electrons. Okay, so at complex 2, we can drive superoxide production. All of those NADHs are dumping their electrons into complex 1, and this green arrow, the electrons are flowing to coenzyme Q, and from there onto the rest of the electron transport chain to make ATP. And what happens, electrons via an FAD input here, and those also go straight into coenzyme Q. And the thing is, if you've got a lot of NA, this can get overwhelmed. This coenzyme Q can get overwhelmed. This is kind of the bottleneck of the electron flow. And if this gets out of the electron transport chain as just an electron, and oxygen minus, it has a negative charge because it's picked up another electron, and those electrons have a negative charge. And so the magic happens here. And so your mitochondrial enzymes are working at full speed. What happens is if you have a lot of FAD inputs or complex 2 inputs, you produce a lot of superoxide, right? Built into our DNA, as it were. We make this enzyme called superoxide, hydrogen peroxide, which is an oxidant, but it's no longer a free radical. The hydrogen peroxide uses something called glutathione, the glutathione system, and there's an enzyme called glutathione peroxidase. It oxidizes the glutathione, and in turn it reduces the hydrogen peroxide. So it's the reduced form here. It has the electrons as GSH. It gives those electrons to hydrogen peroxide so it can become water. It in turn becomes oxidized. It's lost the electrons here, and it gets more electrons from something called NADPH via the enzyme glutathione reductase. So glutathione reductase is taking electrons from NADPH and giving them to oxidized glutathione so it can again become reduced. So I'll break it down another way at the end, and hopefully one of the ways I explain it will make sense. And so ultimately, there's this enzyme called nicotinamide nucleotide transhydrogenase, also known as NNT. And NNT is really crucial to understand, which again, that comes from your food. That comes from the Krebs cycle. And it uses it, the glutathione becomes oxidized. And to get rid of more hydrogen peroxide, you need to reduce the glutathione back to reduced glutathione and pH. But NADPH has to be constantly replenished to continually get rid of the hydrogen peroxide. And where do the electrons come from? Well, they come from, so the NADH is these electrons are moving up and down to NADPH and through reduced glutathione plus. And NAD plus, as I said, is what's required to keep our metabolic rate going, because it takes 3 NAD plus to burn each molecule of acetyl-CoA. Acetyl-CoA is made from fat and carbohydrates, and that's how we get energy to get rid of this hydrogen peroxide. And so, if we follow the electrons, here's what's happening, right? So, as we make these inputs to complex 2, as we drive superoxide, that took an electron. That took calories out of our food, right? The electrons are the calories. And so this electron came back out the quote wrong way. It's not making ATP. We're not using it for energy. We're just, and then it's actually picking up more electrons here to become water. And where did those electrons come from? Well, those electrons also came from the calories in our food. Those become NADH, right? And so, those electrons from NADH, NNT converts them to NADPH, the water. And so, what you see happening here, this is thermogenic, the way that our body can actually increase its metabolic rate to deal with the fact that perhaps, we ate too many calories, or we have too much stored fat, or whatever. And this is all driven by superoxide production in the first place. And it's driven by our antioxidant system, and it's driven by this enzyme NNT, right? And so cycling, they call it a continuous cycling redox circuit. I'm calling it giving off heat without doing any work. So you're literally just blowing off extra calories as heat. That's what's happening in this photo. And so this is the paper that I mentioned. They studied this in great detail and found that the great majority of hydrogen peroxide produced is reduced. Furthermore, the rate of electron flux through redox buffering circuits, just to say that the more hydrogen peroxide is produced and eliminated, the higher the metabolic rate is. The same, but this one they engineered an extra NNT. So this is the enzyme that replenishes NAD plus and restores your NADPH so that you can continue to eliminate hydrogen peroxide. And this cell culture is engineered to have less NNT, and this is the amount of oxygen that they consume over time. So you can see that the more NNT action there is, the more you can convert superoxide to NAD+, and the higher your metabolic rate will be, because oxygen consumption is a reflection of your metabolic rate. And if you have less of this NNT, your metabolic rate is going to be lower. This is from that same study, and you can see the cells that have extra NNT have much, much higher NAD plus levels, and NAD plus allows you to continue the Krebs cycle forward at a fast rate and burn your food, right? Or burn your calories. And SIRT3 activity, we're going to come back to what the sirtuins are and what SIRT3 is. They're very important in all of this thing, but I'm not going to linger on it right now, other than to say that in the cells with extra NNT, they have high SIRT3 activity. And in the cells that have less NNT, you have lower NAD+, and you have lower SIRT3 activity. This is another paper by that same group that showed the continuously cycling redox circuit. These are mice. So these are the two most common strains of mice that are used in experiments in the lab. The 6J ones come from Jackson Labs in Bar Harbor, Maine. And those mice are genetically, they're the same as this. It's like, but they have this mutation, and they have a mutation in their NNT gene. Other than that, these two strains of mice are pretty much identical. They're very inbred, and they're from the same line. But these ones with the mutation that don't have functional NNT, the light cycle, mice are nocturnal, so they sleep during the day. And the ones with the NNT mutation have a lower metabolic rate during the day. And they also have a lower metabolic rate at night, slightly, although it wasn't statistically significant. And the result of this is that the 6J mice with a slightly lower metabolic rate have nearly twice the body fat composition as 6N mice with a functional NNT gene. And so this tiny bit of metabolic rate change has real world differences, right? And also the mice that don't have a functional NNT are glucose intolerant. So these mice were given an oral glucose tolerance test, and the ones without NNT do much worse at it. And so fat saturation, interestingly, controls ROS production. We'll get to the mechanism of that, but this graph is just to show that that's true. So this is in cell culture, and what happens is these white bars are cells that have given a certain amount of glucose, and they're given palmitic acid, which is a 16 carbon saturated fat, and so you can see that compared to, and so these black bars are the controls, and they just have glucose in this first one. And so you can see that compared to cells that get only glucose, cells that get pure saturated fat, which is something that is not found in nature. You never find pure saturated fat in nature. This is a totally arbitrary lab generated thing. And they say in the study, oh, saturated fat causes mitochondrial dysfunction, because this is probably too much superoxide to deal with all at once, but this is literally never seen in nature. So like the idea that they're saying saturated fat causes mitochondrial dysfunction is kind of silly. But that happens in the literature all the time. And so this is oleic acid down here. So this is a monounsaturated fat. And so what they're doing is in each of these bars, there's a little more and a little more and a little more and a little more monounsaturated fat until finally you see this level of superoxide production and buildup starts to drop. And interestingly, the point at which it starts to drop is when you have even amounts. So you have a 1-to-1 ratio of saturated fat to monounsaturated fat. And as monounsaturated fat starts to outnumber the saturated fat, the amount of ROS production drops even more. So the more unsaturated you are, the less superoxide that you drive, and therefore, due to the NNT system, presumably the less NAD plus you will have. And here's why that is. So this is beta oxidation. This is how we break down our fats. And so this is a saturated fat. You see the two carbons have a single bond between them, and they have two hydrogens on either side of them. And when we say saturated fat, what we actually mean is that they're saturated with hydrogens, right? So this is a fat with a double bond, and you see we lose two of the hydrogens. So there's not as many hydrogens as there can be, and therefore the fat is unsaturated. But the thing is, these two hydrogens and the electrons and the bonds between them are the things that get passed into the electron transport chain as an FAD input. And as we saw, the FAD inputs are the things that drive superoxide production. And so if there are no electrons, if you already have a double bond here, there aren't these electrons to pass into FAD simply don't exist. And so you don't, so every time there's a double bond, you don't have this FAD input. And as I showed again, the FAD inputs are the thing that drive this bottleneck and the things that drive that reverse electron flow. And so again, the more the more double bonds there are, the less superoxide is produced. This is real. And so these are mice that cannot, they lack the enzyme to make monounsaturated fat. And so these mice have very highly saturated fat, especially if they're given a low-fat chow diet is what they call it in the lab. This is a low-fat mouse diet. And so what happens is these mice are forced to make their own fat, right? They don't, there's no fat in their diet. And so they have to make their own fat, and they don't have the gene to make unsaturated fat. So their body fat becomes very saturated. And these mice here, the SCD1 minus, these mice are mice on a low-fat diet that can't make unsaturated fat, and their NAD plus levels are nearly 4 times the amount of normal mice, and their metabolic rate is 40% higher than a normal mouse. And this is just because they can't make unsaturated fat, right? And interestingly, these are high fat fed mice. So these mice here in this column with less NAD plus, so they also can't unsaturate their own fat, but they're being fed 60% of calories as fat. And this is commercial lard, which is high in monounsaturated fat. It's about 16% polyunsaturated fat, and they're getting an additional 2 to 3% of calories, something like that, as soybean oil, which is mostly polyunsaturated and monounsaturated. And so they're, when they're burning their body fat, it's very, very saturated, very high metabolic rate. If you give them unsaturated fat, you lose this NAD plus edge. And these mice here are just as telling. So these mice, they have a gene that they overexpress SCD1, the gene that unsaturates their fat. And so their fat is going to be very high in monounsaturated fat, and it's going to be low in saturated fat. And even on the low-fat controlled diet, they're still full of monounsaturated fat, and they have low levels of NAD+. Because with only monounsaturated fat, you can't drive a lot of superoxide, and you don't get NAD+ back. That's interesting because the amount of saturated fat in American body fat has plummeted between 1962 and today. And so these are real studies from real humans in America, and you can see saturated fat is down by about 50%. Polyunsaturated fat has more than doubled between 1962 and 1991. And probably, if anything, it's only gotten worse since then. And the monounsaturated fat is just holding strong, right? The monounsaturated, probably SCD1 levels are way up, because if you add all this polyunsaturated fat, you expect the other two fats to go down. But in fact, the monounsaturated fat is still being cranked out. And it's the saturated fat in our stored body fat that has gotten absolutely destroyed. And so this is from actually the same study in 1962. They went to a tribe in Africa where they were eating a highly starch-based, highly low-fat diet. And so this is the sort of extreme of human body fat composition. So these guys, starch-eating cultures, had much more saturated body fat than Americans did in 1962. You can see saturated fat goes from 35 or 36 up to over 40, and monounsaturated fat goes from 57% down to 50%. So interestingly, you see this 4 to 5 ratio of saturated fat to monounsaturated fat, which, if you remember, that's exactly what was here. So you see 0.4 millimolar saturated fat to 0.5 millimolar monounsaturated fat. So that tribe in Africa eating mostly starch, in the morning. So, right, they eat starch at night, they burn that starch, they go to sleep, and the next morning they're burning their own stored body fat. And so they're going to be making more superoxide than if they just had glucose and monounsaturated fat available to burn. And they're presumably going to have a high metabolic rate. We can actually look at this. And so these are, there's been lots of metabolic studies where you can look at people's metabolic rate, and these are different groups from the literature. So these are women in America in 1996. They're 23 years old. They're sedentary. And these numbers here, and I apologize, I seem to have lost the graphic, but this is kilocalories burned per day per kilogram of fat-free mass. So these numbers have been adjusted for the amount of lean tissue that these women have. And so these sedentary women in 1996 were at 29 approximately. And another study looked at endurance athletes, also young women, a little older, they're 31. This was also done in America in 1996. Their metabolic rate was 33 kilograms per kilogram of fat-free mass per day. And this is a normal kind of range you see in America. The range I've seen has been between about 26 and 35. So these guys are right on the normal American range. And then they looked at these women in Thailand who were 45 years old, so they're older. They're farmers. It's during rice harvest season. And this is their macros. So they're eating 85% carbohydrates, 10% protein, and 5% fat as calories. And so this is what I mean by a starch eating culture. Like that is a starch eating culture, right? They're getting their calories as starch. And their metabolic rate is a whopping 40.6. And remember that these resting metabolic rates are usually done in the morning. And so would be after these women have an overnight fast and they're burning their own stored body fat, which is presumably going to be very saturated, just like those tribes in Africa. Although I've yet to see a study where they tested people's stored body fat and their metabolic rate and then compared the two, although that would be very interesting to see if there's a correlation. And so what happens now that we're Americans and we have, we've given up our formerly saturated body fat and we've gone very unsaturated, and we did it on purpose. And so what happens? And so the thing that happens is you wind up having acetylated mitochondrial enzymes, and that means that your enzymes are slow and inefficient. And how does that happen? Okay, so here's just an example to show that that's true. Here's our old buddy, the aryl hydrocarbon receptor up in the corner, and we're going to activate him by giving him TCDD, the main activator of, there's lots of activators of him, but this is one that's often used in lab experiments. And you can see that what happens is within 24 hours of having an activated aryl hydrocarbon receptor, cells go into dramatic reductive stress. NAD plus levels drop, right? So we have low NAD plus availability. That is going to result in low sirtuin activity, low SIRT1 activity, low SIRT3 activity. Again, we're going to get to what that means in 1 second. But the end result is acetylated enzymes. And so this is just a specific enzyme. It happens to prevent cancer when it's very active, but if you're in reductive stress, it gets acetylated and turned off. And so the way that this works, the way this acetylation works is, this is my simplified model of the Krebs cycle, right? Fat and carbs are made into acetyl-CoA. They swirl around this Krebs cycle, and as they're doing this, they're converting NAD plus to NADH. And the thing is, they need the NAD plus to go around the cycle, right? And so if you run out of NAD plus, this cycle slows down. And what happens when the cycle slows down is that acetyl-CoA, the thing that feeds into the cycle, increases, right? And so when you look at enzymes, this is like a sort of a 3D picture of this blob-like enzyme, and it has this groove in it, and that's the active site of the enzyme. That's where the enzyme is gonna sort of lock and key model match with whatever its substrate is, and it's gonna catalyze some reaction. And what happens is when acetyl-CoA levels go up, the acetyl groups literally just stick to lysine amino acids in the enzyme, and that process seems to be spontaneous. It seems like we don't actually need an enzyme to make this happen. It seems like the acetyl groups just literally spontaneously stick onto the enzymes, and this happens all the time. But of course, as acetyl-CoA levels go up, you've got more acetyl groups around. It happens more often. And the enzymes that actually come along and they take these acetyl groups off. So the sirtuin family, SIRT1 and SIRT3, are known as NAD plus dependent deacetylases. And so their job is just to take these acetyl groups off, just like a pair of scissors would. But the catch is they can only take the acetyl groups off if they have NAD plus availability. And of course, we know the reason that more acetyl groups were sticking in the first place is we don't have a lot of NAD plus around. Furthermore, as the sirtuins do their job, they convert the NAD plus to something called nicotinamide. So as they're removing the acetyl groups, they're actually adding to the problem of lacking NAD plus. And so you see this downward spiral happening where NAD plus lack in the first place led to high acetyl-CoA, led to more acetylation, led to more need for SIRT1, but the extra use of SIRT1 is continuing that downward spiral of using up the NAD plus supplies. This, again, just to show, NAD plus levels go down, SIRT1 activities drop, right? The NAD plus dependent deacetylase, and when deacetylase activity slows down, acetylation goes up. And so this study looked at aged mice. They looked at the livers of aged mice. And what they find is that a lot of the mitochondrial enzymes in the aged mice become acetylated, but in particular, the enzyme NNT was markedly hyperacetylated in aged livers. And if you remember, NNT is one of the main enzymes responsible for replacing NAD plus levels. And you see again this downward spiral into aceted mitochondrial enzymes, because because the whole problem is diminishing NAD plus levels, and the first enzyme that becomes acetylated and turned off is the very one that replaces NAD plus levels. And furthermore, when the NNT becomes acetylated, that causes oxidative stress. Why does that happen? Well, because remember that NNT, as it's replenishing our NAD+, what it's doing with those electrons is it's providing NADPH. And remember that electron flows, the reduced NADPH, those electrons flow through reduced glutathione, and they reduce hydrogen peroxide back to water. And so the less NADPH you have, the less ability you have to reduce hydrogen peroxide back to water. And at the same time, NAD plus levels are dropping. So your metabolic rate drops, NADPH drops, you can't remove ROS. NADPH levels increase, and this is reductive stress, which of course is the thing that drives oxidative stress. And this is from episode 6. I showed this same graph, but this is lipid peroxidation. So this is oxidative stress in cells that were given TCDD, right? So we activated the aryl hydrocarbon here, receptor here, oxidized lipids increase. But what you can see is they increase slowly for a while, and then they really explode. And the point at which they really explode coincides fairly tightly, or perhaps precedes a little bit, this dropping level of NADPH. So it's possible that these two events are somewhat correlated, and as oxidative stress is increasing, NNT is becoming acetylated, and NADPH levels are dropping. Furthermore, if you look at obese humans and torpid animals, we have highly acetylated mitochondrial enzymes. This is in children, and you can see the obese children have about a little more than double the amount of acetylated complex 1. So that is that's the mitochondrial complex that actually oxidizes NADH to NAD plus and hands those electrons through to create ATP. And of course, if you have acetylated complex 1 and you can't oxidize NADH, this is again adding to that downward spiral of acetylation. You see the torpid animals, so ENT. So this is different parts of the year. This is like summer. SA is summer active, and you can see the acetylation levels are at their lowest. ENT is entrance into torpor, so they're getting ready to hibernate. Acetylation levels are going up. ET is early torpor. So as they become torpid, acetylation levels really explode. And so you see that torpid animals, again, are looking a lot like obese humans. Furthermore, complex 2, the thing where the FAD inputs from saturated fat go, well, to drive those electrons, or drive that superoxide production, they're decreased. This is in women who are pregnant, and these ones are obese and have normal glucose tolerance. And it doesn't matter that they have normal glucose tolerance. The obese women have a 40% reduction in complex 2 activity. These ones have gestational diabetes, but it doesn't matter. All of the obese women have reduced complex 2 activity. These are animals going into torpor, and you can see at early entrance to torpor, the first thing that happens is that animal getting ready to hibernate is reducing complex 2 activity. And this is another paper looking at activity of succinate dehydrogenase, and they conclude that complex 2 is regulated by reversible acetylation, and that SIRT3 can give complex 2 its activity back. And so that just shows us that the reason that complex 2 is deactivated in obese humans and mammals is because of acetylation. Right, reductive stress and acetylation. That's the problem. So what do we do? Well, one thing we can do is we can just stop eating unsaturated fats, because you saw how that affects NAD+ levels. Also, I would argue if you are obese and if you are struggling with reductive stress, at least for now, till you get it under control, stop eating monounsaturated fat too. And that is that's a controversial stance, but to me right now, I'm trying to get my body fat back to that of a normal human being, something more like a starch-eating human being. And so for the time I'm not eating olive oil, I'm avoiding poultry, which also is a source of polyunsaturated fat because of the corn and the soybeans that they're fed. I don't eat commercial lard or commercial pork fat because that's another source of polyunsaturated fat. And I'm not even eating olive oil or avocado oil either. Trust me, you have plenty of stored MUFA. You have no lack of MUFA in your body. And that stored body fat, of course, goes into your mitochondria. And so the other thing we can do is we can do things to oxidize that NAD pool, right? We want to oxidize NADH back to NAD+. Remember, NAD+ is the oxidized version of NAD, and that's the one we want. And so doing this will increase our metabolic rate, lower acetyl-CoA levels. It'll activate SIRT3, and if we have low acetyl-CoA levels and activated sirtuins, we can get our mitochondrial enzymes deacetylated, hopefully. Especially, we want to deacetylate complex 2 and NNT, because complex 2 helps us drive superoxide production when we're not in reductive stress, and NNT helps us convert that superoxide back to NAD+. And so I've talked about all of these things on my blog before. If you've followed my work for a long time, this is a refresher. So I'll go through these first ones kind of quick. I talked about alpha-lipoic acid in the last video. I've talked about pyruvate. These are both super incredibly useful tools. Succinate as well, and stearic acid. I sell Succinate and stearic acid supplements at fireinabottle.net/shop. And if you like these videos, if you like the blog, you buy these products. It, of course, helps me to keep making these videos. All of these things are all natural. They're made in the body. Whoops, that slide's extra. And so alpha-lipoic acid increases NAD+ levels. And so these are mice. I showed this in episode 6, but they made these mice fat. They gave them a little alpha-lipoic acid. They lost some weight. You give them a lot, they become lean. And they also measured in this study NAD+ levels, and you can see that as you increase the amount of alpha-lipoic acid, NAD+ levels go way up, triple. They triple, in fact. And this is in live mice, just fed a supplement of ALA in their diet. So this really works, and this is the acetylation level of their enzymes. It goes way down, right? That's what we want. When I say it's made by humans, I don't mean that the supplement tablet capsules are made by humans in a factory, although that's true. What I mean is we make alpha lipoic acid in each one of our cells in our mitochondria, so it's a totally natural part of humans. You're not going to have any weird reactions to it, hopefully. ALA, so this is in humans. This is a meta-analysis of randomized controlled trials. Alpha lipoic acid does cause weight loss in humans. Not a huge amount on its own, but this is suggestive that it's pushing us in the right direction. This is how it works. Our cells take it in. We consume it in the oxidized form. So this disulfide bridge is the oxidized version of ALA. NADH just automatically, without any enzyme help, reduces it. So that oxidizes the NAD+, right? Because when the ALA gets reduced, the NADH gets oxidized, and it gives us back NAD+. The normal kind goes into the cytosol and into the mitochondria. And the biggest complaint I've had from people who've tried it is they get some fatigue from ALA, and they get some kind of like head swimminess. And I'm sorry I lost, whoops. This is supposed to say RALA. So this is the quote right-handed version of ALA is called RALA. And you can find it if you Google it. It's probably better. Maybe. I've heard some reports back that with RALA, people experience less of this fatigue. It does cost about twice as much. Moving on to pyruvate. So pyruvate is essentially a half molecule of glucose. This is the process of glycolysis. We take glucose into our cells, and we go through glycolysis, and this converts the glucose into two molecules of pyruvate, where they go into the mitochondria and are made into acetyl-CoA. The important step here is they get stripped of an electron pair at this step, and they convert two molecules of NAD+ to NADH. So pyruvate is like the oxidized form of glucose. So in the same way that you consume alpha lipoic acid in the oxidized form, and it gets reduced and that oxidizes NADH, pyruvate does the exact same thing. And pyruvate and lactate in your bloodstream form a redox buffer. And the way this works is you eat a bunch of pyruvate, and you can see there's this enzyme in all of our cells called lactate dehydrogenase. And what it does is it just converts lactate to pyruvate, and it converts pyruvate to lactate. It goes both ways. So if you eat a lot of pyruvate, what happens is you convert a bunch of it over to lactate. And when you convert pyruvate to lactate, NADH becomes NAD+. And our cells are constantly importing and exporting lactate and pyruvate. And since they're constantly bringing it in and out, and this enzyme will keep this pool, the pool inside of the cell, the redox balance, equivalent to the redox balance outside of the cell. And so this is just constantly cycling in all of our cells. And so our whole body in the cytoplasm, the ratio of pyruvate to lactate is equivalent to that in the blood. And if we add to this side of the equation, the pyruvate side of the equation, we will have to oxidize some NADH back to NAD+ to wind up with more lactate. And when I brought this up in previous videos, people were concerned. They said, well, won't lactate build up? And the answer is sort of, but we don't care about that, because lactate is, remember, just the same as glucose. And we can burn glucose just fine. Right? It's not that we can't. It's just that in this case, we got over our skis on the NADH side of the equation, and we're trying to haul it back by adding some pyruvate. Once our mitochondrial enzymes get deacetylated, we'll be able to deal with the NADH just fine. And once we get some of that acetyl-CoA out of our mitochondria, we'll be able to burn that lactate just fine. And so pyruvate oxidizes the NAD pool. This is just a neat example. These are normal human liver cells. They have an NAD+ to NADH ratio of about 400 to 1. And so our cells are in a very oxidized, in the cytoplasm, our NADH pool is normally in a very oxidized form. Cancer cells are in reductive stress. So in these human lung cancer cells, the NAD plus to NADH ratio was all the way down to 200 to 1, which sounds like a lot of NAD plus, but believe it or not, 1 molecule for every 200 molecules of NAD plus, that's too much. That's already reductive stress. But if you give them pure lactate, if you give these cells pure lactate, which again can never happen in nature, this is a complete lab phenomenon, you can drive the NAD plus to NADH ratio all the way down to 33 to 1. And conversely, if you give them pure pyruvate, which again only ever happens in the lab, you can drive that ratio all the way up to 1300 to 1. So this is a very oxidized NAD pool, and we got there by adding more pyruvate than you could ever hope to add yourself in a real live human. And again, pyruvate trials have shown a statistically significant difference in body weight with pyruvate compared to placebo. Pyruvate supplements got a little weird in the late 90s. This guy was ultra promoting them on these infomercials and said a bunch of stuff about them, which was not backed by science. And he got into trouble. And I think as a society, we threw the baby out with the bathwater. I think pyruvate is still useful, even though that guy said made extraordinary claims about it. The key is, much like the ALA, the pyruvate does seem to be pushing us in the right direction. So probably just pyruvate alone is not going to fix you if you keep eating soybean oil, right? And so, you know, we have to think about this as multiple steps towards the end goal. Succinate. So succinate gives its electrons directly to complex 2, right? And that is the thing that drives superoxide production at complex 1, which is the thing that drives NAD plus production at NNT. So I think about ALA and pyruvate as sort of like being on defense. Those are things that help us generate some NAD plus and help us get our mitochondrial enzymes deacetylated. Succinate dehydrogenase, by supplying a flood of electrons right to complex 2, that's like going on offense. It's like, okay, now that we're functioning a little bit better, now that we're running smooth, let's go on offense, right? Let's really drive some caloric burn. So like I say, succinate dehydrogenase is driving the superoxide production here, and we are using that to drive forward this NAD plus. You can see this so this is in mice. So these mice have been given succinate in their food. And this graph is sort of backwards than the way it's been presented. NADH is on the top. And so in this case, the mice given succinate have about 5 times the amount of NAD plus as mice without it. So we're really driving NAD plus with succinate. This is complex 1 activity. You can see it's way up, probably because it's deacetylated. This is complex 2 activity, right? So this is where succinate dehydrogenase, or this is where succinate gives its electrons to succinate dehydrogenase. That's complex 2. Activity is way up. This is athletic performance. The mice given the succinate within 30 minutes have increased grip strength. They can lift more weights. They do all kinds of weird things to this mice to make them hold on to hang from bars and lift weights and stuff. Anyway, and what you see is the lactate content of their muscles drop because the having that extra NAD plus, they can continue to burn that lactate oxidatively rather than having to convert more pyruvate to lactate and store more lactate in their muscles. And lactate is kind of what makes your muscles sore. And those mice given succinate have an increased metabolic rate. And so these are the same mice, same diet. This is over a 36-hour period. You can see these bluey purple squares. Those are mice given succinate in their diet. And they are just simply burning more oxygen 24 hours a day than mice that are not supplemented with succinate. Succinate causes dramatic weight loss in mice. This is after 4 weeks, and I think this metric is percent of body weight. So they've lost 4% of their body weight or something in only 4 weeks, and they have significantly more lean mass and significantly less fat mass than mice on the same diet that did not get Succinate. And again, I'm just starting to sell 800 milligram Succinate capsules at fireinabottle.net/shop. I'm also waiting for mine. I apologize if you ordered them before. There was another 1 week delay, but they should ship next week. And I am planning on starting to take 3 grams of Succinate with Pyruvate probably twice a day, as well as a capsule of RALA. That's my plan that I'm going to trial soon. Stearic acid has the most FAD inputs of any fat, and so this is another thing you can do, right? So you can also add stearic acid to your diet. Stearic acid is the longest, most saturated fat, and so it has the most FAD inputs. So compared to any other fat, stearic acid is going to drive the most superoxide production. And you can see these are mice kept on different diets: a low-fat diet, a corn oil diet, a safflower oil diet. And you can see the stearic acid diet keeps the mice the leanest. This is percent of total body fat. Okay, so the stearic acid fed mice have the leanest bodies of any mice. And again, you can get that at fireinabottle.net/shop. So conclusions. Good superoxide is superoxide produced at complex 2 when we are not in reductive stress, when we do not have too much NADH and not enough NAD+. That was a lot of knots. Good superoxide drives NAD+ production via NNT. So you can see the superoxide is shooting out of complex 2, and it's ultimately being converted to NAD+. NAD+ generation by NNT controls the metabolic rate. Remember, we saw this graph. These guys have more NNT and a higher metabolic rate. Succinate dehydrogenase and NNT, which is complex 2, and NNT become acetylated in obesity and in torpor. You can't drive good superoxide if you have an acetylated complex 2, and you can't make an NAD+ if you have an acetylated NNT. And so what we kind of want to do is get our enzymes deacetylated by oxidizing the NAD pool with alpha lipoic acid, with pyruvate, with stearic acid. And once we've done that, then we can try to drive our good superoxide, NAD+ levels, and metabolic rate with Succinate. Like I say, that is available at fireinabottle.net/shop. The next episode is going to be great. We're going to talk about vitamin B12. We're going to talk about folate. We might even talk about vitamin B6. We'll see if it makes it in. That is really interesting. We're going to talk about all those things and how they interact with the aryl hydrocarbon receptor. I know, shock. I'm going to talk about the aryl hydrocarbon receptor again. Who saw it coming? Come back for that. It's going to be really good.